Hello friends, today we are going to learn about activation energy and transition state, enzyme activity, specific activity, common features of active sites, enzyme specificity and types and theories. Before going into details, let's have a brief introduction to enzymes. Enzyme is a substance that acts as a catalyst in living organisms, regulating the rate at which chemical reactions proceed, without itself being altered in the process. The biological processes that occur within all living organisms are chemical reactions, and most are regulated by enzymes. Without enzymes, many of these reactions would not take place at a perceptible rate. Enzymes catalyze all aspects of cell metabolism. This includes the digestion of food, in which large nutrient molecules such as proteins, carbohydrates, and fats are broken down into smaller molecules, the conservation and transformation of chemical energy, and the construction of cellular macromolecules from smaller precursors. Many inherited human diseases such as albinism and phenylketonuria result from deficiency of particular enzyme. Enzymes also have valuable industrial and medical applications. The fermenting of wine, leavening of bread, curdling of cheese, and brewing of beer have been practiced from earliest times, but not until the 19th century were these reactions understood to be the result of catalytic activity of enzymes. Since then, enzymes have assumed an increasing importance in industrial processes that involve organic chemical reactions. The uses of enzymes in medicine include killing disease-causing microorganisms, promoting wound healing, and diagnosing certain diseases. All enzymes were once thought to be proteins, but since the 1980s, the catalytic ability of certain nucleic acids called ribozymes or catalytic RNAs have been demonstrated, refuting this axiom. Because so little is yet known about the enzymatic function of RNA, this discussion will focus primarily on protein enzymes. A large protein enzyme molecule is composed of one or more amino acid chains called polypeptide chains. The amino acid sequence determines the characteristic folding patterns of the protein's structure, which is essential to enzyme specificity. If the enzyme is subjected to changes such as fluctuation in temperature or pH, the protein structure may lose its integrity and its enzymatic ability. Denaturation is sometimes, but not always, reversible. Now let's understand what is activation energy and transition state. Enzymes are biocatalysts produced by living organisms and are generally proteins in nature. Enzymes are not used up by the chemical reactions where they act as biocatalysts. Energy required to start a reaction is called activation energy. Enzymes basically lower the activation energy by modifying the substrate. The activation energy of a chemical reaction is the amount of energy needed to raise the reactants to the transition state from which the reaction can proceed. An enzyme is a biochemical catalyst which enables reactants to reach the transition state at a lower energy, and in doing so increases the rate of chemical reaction. Reactants and products have specific energies. In order to transform the reactants into products, the reactants would have to go through a transition state which is usually higher in energy. A theory was put forward in 1935 by Henry Eyring, then at Princeton University, and by Meredith Gaviney Evans and Michael Polanyi of the University of Manchester to explain transition state, known as transition state theory. TST, that is transition state theory, is also referred to as activated complex theory, absolute rate theory, and theory of absolute reaction rates. Transition state theory, TST, explains the reaction rates of elementary chemical reactions. The theory assumes a special type of chemical equilibrium, that is quasi-equilibrium, between reactants and activated transition state complexes. TST is primarily used to understand qualitatively how chemical reactions take place. TST has been less successful in its original goal of calculating absolute reaction rate constants because the calculation of absolute reaction rate requires precise knowledge of potential energy surfaces, but it has been successful in calculating the standard enthalpy of activation, the standard entropy of activation, and the standard Gibbs energy of activation for a particular reaction if its reaction constant has been experimentally determined. To get to this transition state, the system requires the activation energy. Finally, the products reduce their energy to arrive to the final product state. By definition, transition state is the transitory of molecular structure in which the molecule is no longer a substrate but not yet a product. All chemical reactions must go through the transition state to form a product from a substrate molecule. The transition state is the state corresponding to the highest energy along the reaction coordinate. It has more free energy in comparison to the substrate or product. Thus, it is the least stable state. The specific form of the transition state depends on the mechanisms of the particular reaction. In the equation, S goes to X goes to P, where X is the transition state. Now, enzyme activity. Enzyme activity is a measure of the catalytic ability, measurement of decrease in substrate concentration in a period of time and increase in the concentration of product after a period of time, or two ways to determine the enzyme activity. Various factors may affect the activity of enzymes by either affecting the frequency of the enzyme substrate collisions or by affecting the capacity for the enzyme and substrate to interact. Example, denaturation, temperature, pH, and substrate concentration will all influence the rate of activity of an enzyme. How temperature affects? Basically, increasing the temperature will increase the speed and motion of both enzyme and substrate, resulting in higher enzyme activity. This is because a higher kinetic energy will result in more frequent collisions between the enzyme and substrates. Now, what about low temperatures? Low temperatures result in insufficient thermal energy for the activation of enzyme catalyzed reactions to proceed in general. At an optimal temperature, which may vary for different enzymes, the rate of enzyme activity will be at its peak. Higher temperatures will cause enzyme stability to decrease as the thermal energy disrupts the enzyme's hydrogen bonds. This causes the enzyme, particularly the active site, to lose its shape, resulting in the loss of activity. Now let's know about effect of pH. Changing the pH will alter the charge of the enzyme, which in turn will alter protein stability and overall shape. Changing the shape or charge of the active site will diminish its ability to bind the substrate, abrogating enzyme function. Enzymes have an optimal pH, which may vary between enzymes, and moving outside this range diminishes enzyme activity. One more important factor is substrate concentration. Increasing substrate concentration will increase the activity of corresponding enzyme. More substrates mean there is an increased chance of enzyme and substrate colliding and reacting within a given period. After a certain point, the rate of activity will cease to rise regardless of any further increases in substrate levels. This is because the environment is saturated with substrate and all enzymes are bound and reacting. Now let's learn about specific activity. Specific activity is a term used to measure the rate of reaction of an enzyme with a substrate. Specific enzyme activity, specific activity for short, is a measure of the purity of an enzyme solution and is quoted as units per milligram. The value becomes larger as an enzyme preparation becomes purer, since the amount of protein is typically less, but the rate of reaction stays the same or may increase due to reduced interference or removal of inhibitors. It is used in protein isolation methods to indicate the percentage of purification and is reported when purchasing enzymes to indicate how much you are getting. It is the amount of product formed by an enzyme in a given amount of time under given conditions per milligram of total protein. The amount of product formed per time is referred to as an enzyme unit and is typically given in micromoles per minute or nanomoles per minute. This is a rate of reaction that is assumed to be proportional to the amount of specific enzyme catalyst. Total protein is expressed as milligrams per ml. Therefore, specific activity is calculated by dividing the number of units per ml by the protein concentration in milligrams per ml to get micromoles per minute per mg. For example, the specific activity of the isolated enzyme was measured at 150 micromoles per minute per milligram protein before purification and 800 micromoles per minute per milligram after purification. Specific activity is an important measure of enzyme purity. Different batches of a pure enzyme should have the same values, and even diluting an enzyme solution many times will have identical specific activity values, even though there will be different enzyme activity values. This is because in calculating specific activity, the numerator and denominator are affected equally. Specific activity is very different from activity, but the calculation of specific activity is still dependent on the activity value. This means that the stated specific activity value also will be dependent on the enzyme unit definition. This was all about activation energy and transition state, enzyme activity, specific activity. Now, let's know about active sites, their features, enzyme specificity, and different theories pertaining to enzyme specificity. Starting with active sites and features of active sites. The active site of an enzyme is the region where substrate molecules and cofactor, if any, bind and undergo a chemical reaction. The active site consists of residues that form temporary bonds with the substrate and residues that catalyze a reaction of that substrate, that is catalytic site. Although the active site is small relative to the whole volume of enzyme, it only occupies 10 to 20% of total volume. It is the most important part of the enzyme as it directly catalyzes the chemical reaction. It usually consists of 3 to 4 amino acids, while other amino acids within the protein are required to maintain the protein tertiary structure of the enzyme. Proteins are made of small units called amino acids, and in enzymes that are proteins, the active site gets its properties from the amino acid it is made of. These amino acids may have side chains that are large or small, acidic or basic, hydrophilic or hydrophobic. The set of amino acids found in the active site, along with their positions in 3D space, give the active site a very specific size, shape, and chemical behavior. An enzyme's active site is uniquely suited to bind a particular target, the enzyme substrate or substrates, and help them undergo a chemical reaction. Now, enzyme specificity. One of the properties of enzymes that makes them so important as diagnostic and research tools is the specificity they exhibit relative to the reactions they catalyze. A few enzymes exhibit absolute specificity, that is, they will catalyze only one particular reaction. Other enzymes will be specific for a particular type of chemical bond or functional group. In general, there are 4 distinct types of specificity. These are absolute specificity. In this case, here, the enzyme will catalyze only one particular reaction. Second, group specificity. Here, the enzyme will act only on molecules that have specific functional groups, such as amino, phosphate, and methyl groups. Linkage specificity. In this case, the enzyme will act on a particular type of chemical bond, regardless of the rest of the molecular structure. And stereochemical specificity. Here, the enzyme will act on a particular steric or optical isomer. Now, theories pertaining to enzyme specificity. The active site has a unique geometric shape that is complementary to the geometric shape of a substrate molecule, similar to the fit of puzzle pieces. This means that enzymes specifically react with only one or a very few similar compounds. Theories that were given pertaining to enzyme specificity are lock and key theory and induced fit theory. Now, lock and key theory. The specific action of an enzyme with a single substrate can be explained using a lock and key analogy first postulated in 1894 by Emil Fischer. In this analogy, the lock is the enzyme and the key is the substrate. Only the correctly sized key, that is substrate, fits into the keyhole, that is active site of the lock, that is enzyme. Smaller keys, larger keys, or incorrectly positioned teeth on keys, that is incorrectly shaped or sized substrate molecules, do not fit into the lock, that is enzyme. Enzymes work like a lock in the chemical reaction that is necessary to maintain life. The body contains many enzymes and many substrates, but for life to continue in a healthy manner, the correct enzyme must efficiently locate and unite with its specific substrate. Each enzyme can attract its specific substrate and accelerate the chemical reaction that must occur in the appropriate time span. Each enzyme has receptor sites that allow specific substrates to enter and create a reaction product. The enzyme sites work like the keyhole in a lock. Like the lock on a door, only certain keys will fit in the keyholes and perhaps only one key will open the lock. Putting the wrong key into the keyhole and you can prevent the correct key from unlocking the door. Each enzyme will only respond to one or two substrates, which work like keys for the enzyme lock. The molecular structure of the substrate must correspond in size and shape to the receptor site of the enzyme to produce the desired chemical response. When the enzyme locates its appropriate substrate, the substrate enters the receptor site and both the enzyme and the substrate transform to create a complete union so the chemical reaction can occur. Another theory is induced fit theory. As we know, not all experimental evidence can be absolutely explained by using the so-called rigid enzyme model assumed by the lock and key theory. For this reason, a modification called the induced fit theory has been proposed. The induced fit theory assumes that the substrate plays a role in determining the final shape of the enzyme and that the enzyme is partially flexible. This explains why certain compounds can bind to the enzyme but do not react, because the enzyme has been distorted too much. Other molecules may be too small to induce the proper alignment and therefore cannot react. Only the proper substrate is capable of inducing the proper alignment in the active site. The induced fit model is actually an offshoot of an earlier theory proposed by Emil Fischer in 1894, the lock and key model. The lock and key model states that the substrate acts as a key to the lock of the active site. The active site and substrate are exact matches for each other, similar to the puzzle pieces fitting together. In this model, only a single substrate is the precise match for the enzyme. Once the enzyme finds its exact counterpart, the chemical reaction can begin. The induced fit model is generally considered the more correct version. This theory maintains that the active site and the substrate are initially not perfect matches for each other. Rather, the substrate induces a change in the enzyme. This is similar to placing your hand in a glove. Getting the first finger in may be difficult, but once you complete this initial step, the glove slides on easily with much less energy because it is now properly aligned for your hand. These were the two theories, or rather two basic theories, put forward regarding enzymes. Thank you